Assalamu alaikum. Right now what we will do is go to a different topic and talk about DNA replication. As always, the main resource is this lecture, and you can refer to Cooper's textbook, chapter 6, about 15, 16 pages. So let's go over some basic information first. If I say the word genome, it means the entire DNA content of a cell. Okay, so I can extract DNA from one cell, for example, and that would be the genome of that cell. Or I can extract DNA from group of cells that are similar, like skin cells, for example, or let's say blood cells or brain nerve cells, and so on. So we're talking about the whole genome of these cells. Well, whether I extract DNA from skin, liver, or nerve cells, it should be the same, virtually the same. Um, so it doesn't matter sort of where you extract DNA from, and that would be the individual's genome. Now we can talk collectively about the human genome, okay, or we can say the bacterial genome or the yeast genome, and so on. Now, in all cells, DNA is organized into chromosomes. And in bacteria, there's only one chromosome, and this chromosome is circular. Now, in eukaryotic cells, the genome, DNA, is organized into multiple linear chromosomes, and usually these are complex with proteins known as histones, and we talked about histones before. These are the positively charged proteins that wrap and package DNA into nuclei. Now, what's really important here is that when cells divide, DNA must be copied. It must be duplicated or it must be replicated. Now, and it has to be accurately replicated, okay? So there should not be any variation, any quote unquote mutation in the DNA. Otherwise, there would be, it might result in a disease or a certain pathological condition. Now, yet, variation is important because that's what makes us different overall. Okay, so sometimes this is what some people say that this is the mode or the mechanism of human evolution. But so variation is important though. Now, the thing is, basic information again, DNA synthesis is carried out by enzymes known as DNA polymerases, and the substrates of these enzymes are deoxyribonucleotides.
Okay, so the thing is, the scientists didn't know how chromosomes are replicated because this is necessary for continuation of life. And they came up with three theories, and they said that basically you have two original chromosomes in black, and you have two daughter cells, okay? And these daughter cells, each one of them would have the two original chromosomes, and one would have two new chromosomes. And they thought that it was dispersive. That is, you have portions of the parental chromosomes and portions of new chromosomes in the same chromosome. But these theories turned out to be wrong. So scientists knew by doing a few experiments that it's actually DNA is replicated in a semi-conservative fashion, meaning that um, one strand is conserved. Okay, so semi means sort of partial uh, uh, conservation of DNA. So one of the chromosomes comes from the parental cell, and one is actually a whole new chromosome. So
Now, when DNA synthesis was visualized first, this is an image of a beautiful experiment that was done in dividing bacterial cell. And what you see in here, what happens is that replication was initiated, and then radioactive substrate was added. I believe it was thymidine, it was added, so the newly added substrate would be integrated into the replicating DNA. And as you can see in here, that DNA synthesis occurs bidirectionally. That is, it starts at a central point right here, and it goes in two opposite direction. So it looks more like a fork. It looks more like a bubble. So that's the replication bubble, and it can be divided. So you can draw a line right here, and it can be divided into two forks. Okay, and these are known as replication forks. Now this here is the parental strand, and again you start at a central point right here and goes in two opposite directions right there. Okay, now the parental strand is also known, by the way, as a template. So right here it's the template. Now this is the template, and this is the newly added, the newly synthesized DNA, whereby you have newly added nucleotides. Now remember, I said before that substrates, which are deoxyribonucleotides, these are added to the 3' end of the newly synthesized DNA. So the 5' end is untouched. Okay, so whenever you want to elongate DNA, synthesize DNA, it has to go through the 3' end. Okay. Now notice something that is really important, and that is the energy of synthesizing DNA comes from the substrate itself. Okay, so right here the substrate it's a triphosphate, like ATP, for example, GTP, TTP, CTP, and all of them are deoxynucleotides. Okay, so whenever the enzyme adds the substrate to the 3' end right here, a pyrophosphate is removed. That is, two phosphate groups are removed, and the third phosphate is the one that forms the phosphodiester bond with the hydroxyl group. Okay, so synthesis goes in one direction, which is 5' all the way to 3'. So whenever you want to elongate DNA, it's elongated at the 3' end. So it continues to go in this direction, in the direction of the 3' end. Now think about it now. Look at the template. How is the template read? Okay, in what direction is the template read? The template has to be anti-parallel to the newly synthesized DNA. So if the newly synthesized DNA right here, you have the 5' end and it goes in the 3' direction, the template is the opposite. It's anti-parallel. So right here you have the 3' end right there, and it's read in the direction of the 5' end. And this directionality is important. Now the thing is that caused a problem. That is, synthesis of DNA goes in a direction of 5' to 3'. So how about the opposite strand? Does it, because the opposite strand, if you look at the parental strand, it's anti-parallel, right? So it's 5' to 3' for the first one, and the complementary strand is 3' to 5'. And when DNA is synthesized, it is synthesized from 5' end to the 3' end. So how about the opposite strand? How is it synthesized? Does it go in the same direction? Does it go in that direction? That is 3' to 5' in terms of synthesis of the newly synthesized DNA. And the answer is no. It also goes in the same direction. That is 5' to 3'. So it goes this way. So we're showing here one half of the bubble, just one fork, one replication fork. Now, if you look at the DNA here in blue, you can see that it is synthesized in this direction, 5' to 3'. Now, what happens is that this DNA polymerase on the opposite strand, it synthesizes DNA from again 5' to 3'. and again, the template is read, same thing, 3' to 5'. Now what happens here is that as this strand keeps going on, being synthesized, it opens up the fork, and this allows for another fragment of DNA to be synthesized. Okay, so now you have two of them, and these two are connected to each other. Now notice that this strand right here keeps going on, opening up the fork even further, allowing for a third DNA fragment to be synthesized, and so on. And this, after its synthesis, it gets connected to this one right here. Now the thing is, this now is called the leading strand, okay, because it's leading the way. On the other hand, the other one is known as the lagging strand. Lagging means that it's late, okay. Why is it lagging behind? Because it waits for the leading strand to open up the fork for it to be synthesized, okay. So it has to wait. So its synthesis is delayed a bit. So that's why it's known as the lagging strand. Now the leading strand is synthesized continuously, and the lagging strand is synthesized discontinuously. Now the scientist who discovered this mechanism was known as Okazaki, and that's why these short fragments, short newly synthesized DNA fragments that form the lagging strand, these are known as Okazaki fragments.
So DNA replication is really a complex process, and it requires a number of molecules to complete it. And
now, one important feature of DNA synthesis, of DNA polymerases specifically, is that they cannot initiate replication de novo. What do we mean by de novo? It's like from scratch. Okay, it cannot start replication by itself. It cannot start synthesizing DNA by itself. It needs help, and this help, it needs like someone or something to stretch its arm for it, and this thing is known as a primer. Okay, so and this primer happens to be actually an RNA molecule, so its substrates are ribonucleotides, not deoxyribonucleotides. So it is an RNA primer, and this RNA primer is synthesized by an enzyme known as a primase. So what this primase does is that it synthesizes a primer that is composed of 3 to 10 nucleotides, okay? And right after its synthesis, now the DNA polymerase can, grab hands with the primer, and it can start synthesizing DNA.
so each leading strand requires one primer, right? Because you need this primer, and the leading strand just keeps on synthesizing DNA. On the other hand, each Okazaki fragment requires a primer of its own.
Here's an exercise that I'd like you to do. So what I've done is that I've shown you images or illustrations of how DNA is synthesized in one fork. Okay. Now what I want you to do is draw how DNA is synthesized on both forks. Okay. So here, what I want you to do is draw the leading strand and the lagging strand, or the Okazaki fragments, right here. Now you can indicate on your own the 3' end and the 5' end of the DNA, so that you can start drawing. Now remember that we're starting replication right here at this point. Okay, so this is the central point, and it goes in two opposite directions.
Now we do need other proteins as well, and these proteins are a helicase, and we need single-strand DNA binding proteins. So why are these needed? A DNA helicase is important for separating the two strands from each other, okay, to allow the DNA polymerase to do DNA synthesis. How about the single-strand DNA binding proteins? These are also known as replication protein A, okay, and okay, but first, DNA helicase separates the two strands from each other. It requires ATP and it's connected with the primase in a complex known as primosome. Now, remember that this is something that is important for proteins and enzymes, is that they tend to form complexes in order to process any mechanism really fast. Okay?
Now, single-strand DNA binding proteins play a really important role. Now, what they do is that they bind tightly to the single-stranded DNA, okay, without covering the bases, so that the DNA polymerase can still read the nucleotide, so that it keeps on adding the complementary nucleotide to the newly synthesized DNA. So it keeps on reading the nucleotides in the template, in the DNA template. Okay. Now, what do these proteins do? What these proteins is that what they do is that they prevent the formation of hairpin structures. This is a hairpin, so it looks something like this. Okay, so that's a hairpin. Okay, so the thing is that you have the DNA polymerase trying to read the template DNA, and if there are complementary strands like palindromic sequences, for example, or something like that, you will have the formation of a hairpin. Now the DNA polymerase stops, it stalls, it doesn't move forward anymore because it thinks that this is double-stranded DNA and it cannot read double-stranded DNA. It cannot move on top of double-stranded DNA, so it stops and DNA replication stops. Now what happens with these single-strand DNA binding proteins is that they cover they cover DNA so that they prevent the formation of these hairpin structures. Now, something else that they do is that they protect single-stranded DNA from being degraded, because when cells see single-stranded DNA, they immediately think that this could be viral DNA. So immediately they jump on top of this DNA and they degrade it. So what single-strand DNA binding proteins do is that they protect these single-strand DNA, telling cells, don't worry, this is mine, this is on me. I know this guy. Don't harm this DNA. Now the other thing is that these proteins also they help helicases in stabilizing the unwound single-stranded conformation. Okay, meaning what? Meaning that it prevents the DNA, since you have the two strands, the parental strand, the template DNA strands, they are complementary to each other. So there's a good chance they might collapse together, forming double-stranded DNA again. So what these do is that they prevent the reformation of double-stranded DNA.
Okay, so in prokaryotes, there are three DNA polymerases. We have DNA polymerase one, we have DNA polymerase three, and we have we have five DNA polymerases: 1, 2, 3, 4, and 5. Now, number 3 is the DNA polymerase that is synthesis for the formation of the newly synthesized DNA, and usually it is a complex with the primosome, which is made of the helicase and the single-strand DNA binding protein, and it forms with this complex what is known as a replisome. Now, DNA polymerase 1 is important for DNA synthesis, all right, and it also has an exonuclease activity. As I said, it removes the primer. Okay, and number 3 is that, as we will learn in next lecture, it's also important in DNA repair. Now, the other DNA polymerases 2, 4, and 5 are important for DNA repair as well. Now, let's look at DNA polymerase 3. This is really a huge protein. It's not a simple protein. And this protein contains many subunits: alpha, beta, gamma, delta, etc. And what's important about it, about these subunits, is that the alpha subunit is the subunit responsible for DNA synthesis. Okay, but there is epsilon subunit, and it has an exonuclease activity. Okay, and this exonuclease activity is different than the one that DNA polymerase one has. Because it goes in the opposite direction. It goes from the 3' to the 5' end. So what does it do exactly?
So I said that DNA synthesis has to be accurate. Now, it's quite amazing that DNA polymerases, when they replicate DNA, they make only one mistake per 10 to the 9th nucleotide read, which is quite amazing if you think about it. Our genome is composed of 3 billion nucleotides, that is 3 times 10 to the 9th nucleotides, base pairs. So just imagine that upon finishing up replicating the human DNA, only 3 mistakes are made, which is really quite amazing. Now the question is, how does that happen? Why is it that fidelity is really high? And there are two reasons for that. The first reason is that DNA polymerases can catalyze the formation of phosphodiester bonds when the right hydrogen bonding forms. So whenever you have a G, for example, and the polymerase attempts to add a, let's say, T, well, what happens is that you have the wrong formation of hydrogen bonds between the two. Okay, so this is one. So it doesn't fit into the enzyme. The substrates do not fit into the enzymes. Okay, so this gives us, or gives cells, an accuracy of one mistake per 100 to 1,000 nucleotides read or synthesized. Okay, which is quite high still. But what's really important is something known as the proofreading mechanism. Now, the proofreading mechanism is an exonuclease activity of the polymerase. So the polymerase has two enzymatic activities. The first one is the polymerase activity, that is the DNA synthesizing activity, and the second activity is the opposite. It is an exonuclease activity. So what is an exonuclease? What does an exonuclease do? It cleaves DNA. It degrades DNA. So it's the opposite of polymerization. But what does it do exactly? It acts at the end of the DNA, exo. Okay, so that's the opposite of endo or indo. So the direction is important as well. Now, DNA polymerization goes from the 5' end to the 3' end. Now, this proofreading mechanism goes into the opposite direction. That is, it goes from the 3' end backward. It goes back to the 5' end. So let's say that the DNA polymerase puts in a G instead of an A. Now, this is wrong. It doesn't fit. It's okay. So these are like Lego blocks. It just doesn't fit. And the DNA polymerase senses that. So what it does is that it goes backward, okay, and it removes this G. So what it does is that's proofreading, okay. So it goes in the opposite direction, 3' to 5'. It removes the G, okay, using its exonuclease activity, and it adds the right complementary nucleotide instead of it.
Okay, so but we have a problem. And the problem is that the DNA helicase separates the two strands from each other. But what happens is that DNA ahead of the DNA polymerase, which is right here, becomes overwound. Okay, it's almost like doing this act right here. You have overwound DNA, which makes it impossible for DNA polymerase to continue with the DNA synthesis. So how is this solved? It is solved by enzymes known as DNA topoisomerases. Okay, and there are two enzymes, topoisomerase 1 and topoisomerase 2. Topoisomerase 1 produces a transient single strand break. So it sort of like creates a swivel. هذا هو السwivel, meaning that it separates the two chains or two strands from each other. So when DNA is rotated, only one portion of it is rotated, and this is not rotated whatsoever. So that prevents the formation of the overwound structure. Okay. So here we have a look at how DNA is replicated. Okay, now, so we have right here we have the primase, and the primase is associated with the helicase, forming the structure of a primosome, right? And this structure can also be complex with the DNA polymerase 3, okay, and forming the replisome. Now, what happens is that you also have DNA polymerase 1. DNA polymerase 1, here you have an Okazaki fragment. What DNA polymerase 1 does is that it removes the primer right here between this newly synthesized DNA and the other DNA ahead of it, the other Okazaki fragment ahead of it, and it fills in the gap, and DNA ligase then comes in and it connects the two strands from each other, okay, with each other. Now, you also have ahead of the replication fork, you have the Topoisomerase 1. Now, there's another.
Now, the question is, where does DNA replication start at? At any point randomly? No. It starts at a specific position known as the origin of replication. So there's only one in bacterial genomes, there's only one origin of replication. This is where the enzyme DNA polymerase starts DNA synthesis. Okay? And this origin of replication was found to contain consensus sequences. So what do we mean by consensus sequences? Now, consensus means that there's agreement, okay? So it just happens to be a certain region that is found almost the same in different bacterial cells, whether we're talking about the same species or different species, but you can see that they share these sequences, and that's why they're known as consensus. So if you discover, for example, an important region in bacterial genome, and you look at genomes of other bacterial species, you may find similar sequences. So you say that, aha, it means that this sequence is important, and that's why evolutionary it has stayed in place. It hasn't changed, because otherwise these pieces would disappear. So these are known as consensus sequences. Now, it just happens that the origin of replication contains two consensus sequences. The first one is known as the 9-mer, and this means that these are regions known as, they contain 9 base pairs that are common, and it just happens to be 9-mer, the binding site of a protein known as the initiator, that is initiator of replication. Specifically, it's known as DNA. So what it does is that this DNA, it binds to these sites right here, okay? And this binding affects another consensus sequence known as the 13-mers, okay? That is, they're composed of 13 base pairs, and these 13-mers are AT rich. That is, they are rich with A's and T's. So why is that? How does DNA, how is DNA replication initiated? What does DNA do exactly?
It does is that it binds to the 9-mers, it forms a structure like here, and it squeezes on DNA, opening up the AT-rich sequences. So now we know why these sequences are AT-rich. so that the interaction between the two strands becomes weak because these sequences are AT rich. Okay, so that's what DNA does. Now when cells see a bubble, they know that it's time to replicate DNA, so the helicase and the single-strand DNA binding proteins, they jump on DNA and they start opening up DNA even further, and DNA polymerase 1 binds, DNA polymerase 3 binds to the replication bubble, and it starts with the DNA synthesis. Now, what happens in bacteria is that synthesis is bidirectional, so it goes in both directions, and until it hits the other side of the DNA of the chromosome, and then the two strands are separated from each other. Now, notice that you have one new strand and one old strand at the end of DNA replication. Well, how about in humans? What does happen in the human genome? The human genome is huge, so one origin of replication is not enough, and as a result, there are multiple origins of replication. And what happens is that eventually, what happens is that the synthesis is also bidirectional, and eventually, these replication bubbles, they meet halfway, and the two strands are separated from each other. Notice that you also have one old strand and one new strand. So DNA replication in eukaryotic cells and in human cells can be finished in a short time.
Well, how about origins of replication in eukaryotic cells? Well, it's more complex, okay, of course, than prokaryotic systems overall. And a lot of studies have been done on yeast cells. So why yeast cells? Because yeast cells are the simplest or among the simplest eukaryotic systems. They are single cell organisms, just like bacteria. They grow like bacteria. They have similar characteristics to bacteria, yet they are eukaryotic systems. They have linear chromosomes. They have chromatins in their DNA. They have shared DNA sequences with human cells, similar mechanisms when it comes to the way cells grow and divide and die as well. So a lot of work has been done on yeast cells in order to understand how human cells function. And in yeast cells, in a specific species in yeast cells, special consensus sequences were found, and they were known as autonomously replicating sequences. Autonomously replicating sequences, or ARS sequences. And these sequences contain 11 base pair ARS consensus sequence, again known as the ACS right here, and the 3 additional elements. Aha, here's the new word, element. Whenever you hear the word element in molecular biology, it means it is a specific sequence in DNA. Okay, so whenever you hear the word element in molecular biology, it means it's a special sequence of DNA. And right here you have 3 elements, and these are known as B1, B2, and B3. And this whole thing is the origin of replication. Now, you have a protein, a complex known as the origin recognition complex. It binds to the ACS element, consensus sequence, and it can also bind to the B1. And this binding results in recruitment of other proteins known as the MCM, and these MCM are helicases as well as other proteins. And what these do is that then they open up DNA, allowing for other proteins to come on DNA and start DNA replication. Well, that's in yeast, and this is a very similar system, similar to bacterial origin of replication. So how about humans? Well, things are really complex. We don't know yet how DNA is replicated. We know that it starts at certain regions. We don't know why, because there are no elements, there are no consensus sequences, and it is thought so far that proteins recognize origins of replications according to the chromatin structure, the way the DNA is complex with proteins and the way it looks like in three-dimensional space. Okay, so this is really important, the structure, and it's very complex if you think about it. So there are no specific sequences in human DNA like origins of replication in bacteria or this simply yeast cells.
Now, the thing is, the problem is whether we're talking about bacterial DNA or we're talking about eukaryotic DNA or human DNA, what happens is that the chromosomes become tangled up, okay? They become tangled up and they must be separated from each other. And that's why we need the other topoisomerase, which is known as DNA topoisomerase 2, and it's also known as gyrase in bacteria. It requires ATP, which is different than topoisomerase 1. Topoisomerase 1 does not need ATP. That's one difference. Another difference is that DNA topoisomerase 1 creates a single cut in one of the strands, not both of them. On the other hand, DNA topoisomerase 2 what it does is that it creates two cuts on the same strand. But eventually what it does is that it separates the two strands from each other. Now, because DNA topoisomerase 2 is important for DNA replication, for DNA division, inhibiting topoisomerases, whether one or two, can prevent cells from dividing as a result of blocking DNA replication. And as a result, topoisomerase inhibitors are used in the treatment of cancer to stop DNA synthesis and cell growth and proliferation. Now, there's another protein that is needed, and it's known as Pcna, or proliferating cell nuclear antigen. And this protein, what it does is that it guides the DNA polymerases to the primers. Okay, so it's needed for DNA replication, and Pcna is used in diagnosing cancer, so it is a marker of cancer. Okay, why is that? Because cancer cells tend to do a lot of DNA replication and cell division, and as a result, they increase the synthesis of Pcna. And this is shown here by a technique known as immunohistochemistry. So this is done on tissue sections. Okay, so right here, these are normal cells, and what you see staining in brown right here, this is staining of Pcna, and you can see the brown color is a lot in these cancer cells versus normal cells. Okay. So DNA polymerases in eukaryotes are multiple, like in bacteria, but there are more of them. Okay, but I'll focus on 4 of these DNA polymerases, and these are DNA polymerase alpha, delta, epsilon, and gamma. Starting with DNA polymerase gamma, it's a mitochondrial DNA polymerase. Okay, it does have an exonuclease activity, so it is accurate, which is also known as the proofreading mechanism. Okay, now we have DNA polymerase alpha, delta, and epsilon. Now, alpha it has a primase associated with it. It has high fidelity, so it doesn't create that many mistakes. And probably that's because it has high processivity. That is meaning that it is a slow enzyme, so it doesn't make that many mistakes. On the other hand, DNA polymerases delta and epsilon both have high processivity, they're fast, they have high fidelity, they do not create that many mistakes, and probably because they have this proofreading mechanism, they have 3' to 5' exonuclease activity. So what happens is the following. So specifically, DNA polymerase epsilon is responsible for synthesizing the leading strand. Okay, now DNA polymerase alpha, as I said, is associated with a DNA primase. So the primase creates the RNA primer, and the polymerase starts synthesizing the Okazaki fragment. Okay, the beginning of the Okazaki fragment, but then it falls off. Okay, and DNA polymerase delta jumps on DNA, and it continues with the DNA synthesis. Okay. Now, both DNA polymerases epsilon and delta are associated with Pcna, so they are guided by this protein. Now, what happens is that DNA polymerase delta, as it synthesizes the Okazaki fragment, it hits the primer in the next Okazaki fragment, and it needs special enzymes to remove this primer, so it does not do this by itself. It needs help. Okay, and then but it fills in the gap created by these enzymes. Okay, so that's the idea. We have polymerase epsilon, DNA polymerase epsilon is responsible for synthesizing the leading strand. Each Okazaki fragment requires DNA polymerase alpha followed by DNA polymerase delta. Now the thing with eukaryotic DNA is that we have a problem with chromatin. DNA polymerase cannot synthesize DNA until DNA is free of histones. And what happens is that these histones are removed by proteins known as chromatin remodeling proteins. So they remove histones so that DNA polymerase can continue with the DNA synthesis. Now the same old histones as well as new histones can be used to refold DNA in behind the DNA polymerases, and this is done by chromatin assembly factors.
Now, there is a problem when synthesizing linear DNA. And the problem happens to be in the lagging strand. Because right here, let's say you have a point of initiation of replicating linear DNA. That's an origin of replication. So you have primers in red. Okay. Now there's no problem with the leading strand because it keeps on going on, okay, until it gets to the 3' end. Okay. Now the problem happens to be with the lagging strand. So right here you have a lagging strand that is being synthesized right here. Now what happens is that same thing on the opposite strand, okay? Now what happens is that this primer is removed and there is no place whereby DNA, whereby a new RNA primer is added. Okay, so the primase sits right here, or it tries to sit here and tries to synthesize the primer, but it cannot. There is no more space anymore. So what happens is this is a problem with telomeres. Okay, and telomeres are the ends of chromosomes, as you can see in here. Okay, so the ends of chromosomes are labeled in yellow, as you can see in here. Okay, so the problem here is with the telomeric ends of chromosomes.
Okay, so right here what happens again is that when you have this right here and it's not completed and you have another round of DNA replication, when this is removed, this becomes actually shorter. So one of the chromosomes becomes shorter right here. Okay, now the thing is, the end of the chromosome is really important to which is known as the telomere, is really important for stabilizing DNA. Okay, and this is completed, the lagging strand is completed by an enzyme known as a telomerase. Okay, and this telomerase is known as a ribonucleoprotein, meaning that it is a protein that is made of protein plus RNA, and that's why it's known as a ribonucleoprotein. Now, what is this RNA? It functions as a template. Okay, so it functions as a template. So what the telomerase does is that the RNA itself can hybridize to the end of the DNA. Okay, and what happens is that the telomerase elongates the template of the lagging strand. Okay, so it continues with the DNA synthesis. It makes it longer. Okay, it keeps on adding the same repeat, the same sequences, and that's why telomeres, telom by the way means end. Okay, so telomeres are composed of repetitive sequences of DNA. Okay, so this is what it actually does. It keeps on adding, it uses the RNA that is associated with it to elongate the template of the lagging strand. Okay, it keeps on adding the same exact sequence over and over again. Okay, so let's look at this animation right here. Notice that DNA synthesis continues until it gets to the end of the template of the leading strand. Now the other thing is that with the lagging strand, with the template of the lagging strand, this is the primer, but when it's removed, there is no space to fill in this gap right here. Okay. Now the telomerase comes in, and what it does is that it uses its the RNA that is associated with it, and it elongates the strand. That is the template of the lagging strand. Pay attention to something here. There are two mistakes in this animation. Okay, I want you to find out what these two mistakes are. One of the mistakes is in the type of DNA polymerase that synthesizes DNA, and the other mistake is with the RNA molecule that is associated with the telomerase. Okay.
Now there's something fascinating about telomerases, and that is in germline cells, that is in egg and sperm cells, in differentiating, in stem cells, replicating cells, ever replicating cells. These cells have large quantities of telomerases, and they have really active telomerases. The same is not true when it comes to somatic cells. And somatic cells are cells that, just like differentiated cells, like, for example, skin cells, blood cells, liver cells, and so on. Now, these cells do not have high levels of telomerases, and that disables going indefinitely with cell division. So with germline cells and stem cells, they keep on dividing. And telomerases, are still high in number and activity, or quantity and activity, but somatic cells do not have this luxury. So what happens with stem cells is that they keep the chromosomes are kept in the same length. Okay, so it's constant. But for somatic cells, the telomeres get shorter and shorter with every cell division, and that leads to what is known as cell senescence. That is, they stop growing, they stop reacting to the environment, they just stall, they do nothing. Okay, they just sit there with very low metabolism, they cannot divide, they cannot function, they cannot do anything, they just sit there, or they eventually die. Okay, now what happens with cancer cells like melanomas are skin cancers, is that they express high levels of telomerases, and that allows them to divide continuously, indefinitely. They do not die. Okay, so this is a a feature of cancer cells overall, particularly in skin cells, and that is they keep on dividing, okay? Because they express high quantities and very active telomerases. And there was a correlation that was found between the activity and amount of telomerases with formation of tumors, which can lead or evolve or progress to cancer cells. Okay, and that is the higher the activity of telomerases, the higher the quantity of telomerases, the more aggressive cells, the larger the tumors are, and the higher probability that these tumors would progress to cancer. Well, a sad fact of life is that we age and we die. And it is thought to be because of a reduction in the activity and quantity of telomerases that our cells keep on dying, and eventually we have organ failure, and we die. Okay. Now, something else related to telomerases is the sheep, Dolly. I'm sure you've heard of Dolly. Dolly was a cloned sheep, okay? And this Dolly was cloned from another sheep. So it's DNA of another sheep taken into an egg, and this egg then was fertilized, and it produced. Actually, it wasn't fertilized. This DNA was transferred. It was already deployed, transferred to another egg, and the egg was stimulated to as an embryo to become Dolly. Okay, and the thing with Dolly is really fascinating. This is how it was done. So a donor cell was taken from a sheep, another sheep. Okay, and it was then this cell was taken actually from the mammary gland. That is the gland that produces milk when the sheep, gives birth to a lamb. So an egg was taken from another sheep, and the nucleus was removed, and then cells were fused. So now you have the nucleus of this cell into the egg of the other sheep, and the egg got stimulated, and it produced an embryo, and this embryo became Dolly, the sheep. Okay, actually, it was transferred to a to another sheep, a foster mother, and it gave birth to Dolly. Now the thing about Dolly is that it lived for 6.5 years instead of the normal 11 to 12 years of sheep. Okay, not only that, but Dolly looked old early on, and she had certain diseases, okay, like sheep diseases, like sheep arthritis, okay, which was really fascinating, and other things as well. And she looked, if I can say she looked old as well. And the reason for that is that the DNA that was taken, the cell that was taken from the donor sheep, was actually 6 years old. Quite amazing. Okay, so 6 plus 6, that's 12. Okay, so the thing is, when Dolly was born, she was 6 years old, molecularly speaking, because now the chromosomes in these cells are 6 years old. They were shorter than normal. Okay, as you know, if like DNA in this egg. So that's amazing. But science progressed and advanced, and sheep were born at in good health. Amazing.